there's been a question of when exactly the exam will be held, and it will be held actually a week from Friday in class. It will have 70 points worth of questions on it, and that will be multiple choice, short answer. Friday of this week, I will send out 3 essay questions. You have a week to choose one of them, answer it, and upload the answer onto Canvas. And those will be due 9 o'clock on a week from Friday, so right before the exam. And then for the graduate students, I'm not going to change things on you guys, don't worry. Your final reports are due by 5 p.m. of Friday of finals week. So, okay. So, any other questions? Again, please. Yeah.
Did you want the essays posted on Canvas or in a Word document?
No, posted on Canvas. And so I'll set up a place on Canvas where you can upload those. So.
How extensive do our answers for those have to be?
Well, since you have to work on them. Yeah.
Okay.
So, yeah.
So like the extra credit assignment was, but do you want outside sources as well? Okay.
Yep. So that would be great. Okay. Let's see. So, any other questions? Sorry. Okay. So.
Nice.
Today.
Oh, I can wear black shoes.
Oh,
That's funny. I
Okay.
did not even use
Okay, so we've got paper presentation, Ben's presentation, Josh's presentation. So I am going to set my timer. So you have 10 minutes to go over your paper. You each have 15 minutes to go over your slides. And then hopefully we'll still have some time for questions. So with that, Lorena, here is a pointer if you want to use it, but you don't have to. But there you go.
Okay, so this paper is essentially using an Antarctic fungus to do bioremediation of dye. I didn't know about dye being an issue at all in like pollution, but it is a really huge issue. It hinders the light penetration into ocean waters, into wastewaters, kills off plants, lowers oxygen concentrations, that sort of thing. So it is something we really need to solve. It can also cause cancer and be toxic. So, lots of stuff. This study essentially found 2 fungi, which is Lamai, L-M-A-I, 2400 and 2402, that degraded dye tested, which was sulfur indigo blue, greater than 90%. They're both penicillin. I don't know, I'm trying to make this sound interesting. It was kind of a dry paper to read, in my opinion. I don't know why I chose it. They looked at Antarctica for extremophile purposes. So Antarctica is obviously a really extreme environment, really dry, lots of freeze-thaw cycles, really, really cold. Because of that, they're looking there to see if there's just bacteria, fungi that can do different things. Penicillin in particular and cold-adapted fungi are useful for dye bioremediation because they make oxireductases to reduce this dye, specifically MNP, LIP, and lacase that decolorize it. So basically when they're bioremediating this dye and decolorizing it, they're kind of breaking it down so that it does allow light through. I think I'm going to try and just go through thought I had 15 minutes, so I'm trying to rush through it a little bit. With figures more so, they took samples from a couple of different places. They did surface sampling as well as pore sampling for each of them. And this is all in the sediment of the ocean. Yeah, these are the fungi found in different places. Both of the ones that they liked were from the same spot, both of the most effective ones, which I thought was interesting. The others really weren't all that effective by remediating the dye. And then this is the main figure for the paper. So essentially what they're showing is that while some of the different fungi can do different things, they also limit it to filamentous fungi. They got some bacterial samples and things, but they just limited it to this. Like I said earlier, 2400 and 2402 are both the most effective in both of the different temperature cases they tested at. They tested at 15 and 28 degrees Celsius. After they figured out what was the most effective, they kind of did some characterization studies to a certain extent, like how much biomass can they create, what are the optimal temperatures, what's the optimal salinity, that sort of thing. Part of this was also to see if they could draw a correlation between the dye biodegradation and their biomass. They couldn't really, while the two that did degrade the dye better did grow more, there's not that much they can say about it. Would have been really cool if they could prove it to show that that was like the main thing they were using. Like I mentioned earlier, this dye can also be toxic. However, degrading it can make it more toxic to a certain extent, more toxic to intermediates. So one of the things they did study was just using the two they thought were the most effective. How toxic are they? How can they change the dye? So the control, I believe, was just water and the dye. They used cucumber seeds, which didn't seem that optimal considering they're looking at like aquatic plants and things. So they used cucumber seeds and saw how much the roots grew, determining that it kind of does have toxic intermediates. It doesn't do that great of a job, as you can see. This guy here, it's more toxic than the control. At 28 degrees Celsius, again, at 15, everything's pretty much okay. They also, again, did some characterization stuff. Both of the organisms were penicillin, and they were both very closely related, as you would expect, they're from the same spot. They did genetic analysis of these, compared it. Yeah, they just used a bunch of different websites and sources and things to sequence them. Well, got them sequenced, compared them, made a family tree, and then they looked at them under the microscope to do different things. They didn't have any pictures of them, unfortunately. Said that they were like velvety and orange or something. Yeah. So basically, what they found: 29 microorganisms isolated, only 11 were filamentous fungi. 2 are the best decolorizers at both of the different temperatures. Biomass was close to both temperatures. It didn't change all that much. Highest decolorizers did have the most biomass. They probably do degrade it enzymatically. There are 2 different types of degradation. Enzymatically is the one that can have more intermediates, more toxic intermediates, because of the increased toxicity. I think this is what they're doing. So we can look into more. I do have other stuff. How many do we have? I practiced it.
You've got 3 minutes.
3 minutes, okay. Are there any questions? Or I can go through like the methods of how they analyze things and how they isolate things.
Actually, methods and stuff like that, already gone over some of that in class. So I guess one of the questions I have for you is, okay, so why Antarctica? Why find cyclophilic organisms with sort of degradation?
So the cyclophilic organisms were looked at specifically because they've been proven in the past to be able to degrade dyes and things like this. The enzymes that I mentioned, yeah, lytic enzymes, so basically enzymes they create to degrade lipid. Some different types of these, the lipid is pretty close to the dye that they're using. So we were trying to find
Another two to remember is, again, you're probably thinking about, well, one, possibly using these in the environment, but then also these that will have bioreactors to degrade these, or especially the waste that's coming out of these plants or areas where the industry is using a lot of these dyes, then you can do it at lower temperatures, ergo lower costs. So. Anything else? Any other questions? Oh, thanks, Brad. Thank you. That's all great.
Hi everybody, I'm going to be talking about my asset proposal, and it's basically using fungal species to remediate petroleum contaminated soils. So a bit of background, it's obviously very common to see petroleum contaminated soils across the planet. It can happen a variety of ways, just by accidental leakage, intentional leakage. So it's not uncommon to see, and they can cause some serious health effects for humans, including some cancers, and just respiratory damage, nerve damage, things like that from prolonged exposure. This is a picture from Kuwait of just a contaminated field with oil. They're currently working on treating it. And so that's primarily where I was thinking for my experimental site. So a bit of background about Kuwait actually is that it was invaded by Iraq in 1990, and when Saddam Hussein was basically retreating back to Iraq and the U.S.-led forces were entering Kuwait, they tried to hinder them any way they could. They lit some oil fields on fire. They spilled about 580 million gallons of oil into the Persian Gulf. So it's a pretty bad situation environmentally. And so obviously it took a little bit for everything to get stabilized in Kuwait. And once they did in early 2000, they kind of started cleaning everything up. And so they estimated that there were about 252 million gallons that were burned in the oil fields. And it was mostly just in this one, from what I could tell, the Greater Bergen one. And so obviously it's one of the bigger ones, and so that's a huge issue there. So that's primarily where I was thinking for this proposal would be, since that is the most likely type. So my hypothesis here is that we would have some petroleum degradation, and this species of fungi, Aspergillus flavus, has been directly cited as being able to degrade about 80% of the petroleum in an experimental sample in 30 days. So that was something that kind of got me working on that, but I didn't want to focus too much on one species because you can't really select for one species in the environment. You have to work with everything you got there. And so I kind of changed it just to fungi instead. So the other hypothesis is that we would have overall contamination levels decrease due to the coordinated efforts of the fungi to degrade it by competition between each other. So my experimental here, there's a paper that, there were a couple papers I read that had done something similar, but they had done it in other places like in Nigeria and other places that had heavy soil contamination with petroleum hydrocarbons. But they basically took soil samples, and what we would do here is take soil samples about 5 to 10 centimeters deep. That's deep enough that the petroleum is still contaminating it, but not deep enough that it is not clean soil essentially. And we would take it from all around that Greater Bergen oil field just to kind of get representative samples. It's a very big area, so we want to take samples and basically break it into different areas. And then we also want to take a groundwater sample as close as we could to the place that we got our soil sample, try and add that to our media, which I was going to go later. And then we also take a petroleum sample from the closest well, and that is also used in the media from the papers that I took. And these are two light microscope pictures of species, we have Aspergillus flavus again, and then the penicillin. And these are members of the Ascomycota, which are very common in, are the most common in petroleum contaminated soils. And the reason why that people think that is because they think they're more tolerant to high levels of petroleum contamination. So that's kind of the reasoning why they would be more prevalent. And so after we would get our samples, we would take them back to the lab. We would use 5 grams of soil sample and then a mil of our water sample, kind of added to a 9 milliliter saline solution, and that would give us a total of 10 mils. We would dilute it some, and then we would plate it on bigger dextrose agar, which these are actually Aspergillus flavus and another penicillin that have been plated and left to grow. But then we would incubate it for 2 to 3 days in like a 80-degree incubator, just to kind of let things grow. And then after that, we would count on 20 units, just to kind of get that representative of the population that would grow. And then we would also have to subculture to try and get out the bacteria. We could use antibiotics to try and get rid of them, or just selective media to the bacteria. And then after those 3 days of growth, we would take basically a mil of our sample and put it into a cuvette and use a UV spec to kind of get some turbidity measurements, and that would help us track the degradation. And then after that, we would use oil broth, which I didn't know what that was, and I had to look it up. But in the paper, they said that it was basically a mineral salt media, and then they had it at a 1% concentration of petroleum. So that's where that sample of petroleum oil well comes from. So that way it's kind of local to where we got our samples from. And then we would let that incubate for 2 to 3 days again at room temperature this time, get more growth and everything. And then after that, we would take a sample and use GC-MS, and we would run it through there to try and also help track how much petroleum degradation happened. And then this is our expected results. These two charts are actually from one of the papers that tracked the degradation. They only used Aspergillus flavus in their paper, but it's kind of a little bit of an idea so you can visualize the degradation amounts. But our expected results, we would see between 16 to 80% degradation. And that is not just representative of Aspergillus flavus. So obviously we have some competition between the species, and it might lower some for the individual species, but overall that would be it, because they're working towards our end goal, common goal type thing, where they don't know it, but they're helping us by degrading petroleum. And so you can see here this is the Aspergillus flavus. It has a significantly lower abundance of petroleum versus just the control, which didn't have any. They had autoclaved it to make sure that there were no bacteria or fungi or any other microbes in there. So it has had a pretty significant difference after just 16 days. And then obviously we could do genomic testing too, just to see what species we have there, and other things to help identify the exact species. We're using that selective media to kind of figure it out if we wanted to design it a little bit more intentionally.
I just really on that last slide, you mentioned autoclaving. What is autoclaving?
So it's a gigantic machine that you put your samples into, and it raises the pressure and temperature, and it basically kills any bacteria or anything living in your samples without damaging whatever's in there. I don't know if it's like I know describing.
Right. Yeah.
As a biologist, it's really hard to describe autoclaving to someone who's not autoclaving. It's like,
Essentially what you're doing is it's a one-off things with both pressure and temperature. So
it's like how you denature proteins and stuff with it boiling, but some things can't be killed by boiling because they form endospores and can survive high heat. It's really like,
yes.
yeah,
That's exactly.
It's a canner.
Yes.
It's okay, cool.
Very fancy canner. Let's see, any other questions? So, one, do you notice this of the contamination levels still is there around those wells?
Anything I read, there's still like continuing on, so it's still not great, which makes sense to give up.
So, in your report, yeah, find out, this is, I agree with you, I suspect that this is still a very, very large problem that needs to be addressed. My next question is, how did you choose the media that you did to grow the isolates?
There's kind of common and also provides them everything they need just to grow. But also that is like they can use that in several figures. And they go like this is really important for all that. So
Because I think one of the things that you'll have to do is really distinguish, and you're right, that is used very commonly for fungal growth and all. And it's, specific for fungi as opposed to bacteria. And so, because one of the things that you're forming is bacterial growth, focusing on fungi. So, and then you said something about growing these things in an 80-degree incubator?
yes. So they said
Is
that
that what,
80 Fahrenheit.
okay,
28 Celsius.
Celsius?
Yes.
Because if it's 80 degrees Celsius, well then, you're also
Yeah.
killing off your organisms. So you have to be very precise and actually, NASA actually destroyed something that was sent up to Mars because they would start Fahrenheit and Celsius or actually English and metric. So always use metric in science. So, and then I think another thing that we really need to do for your report, so you reference the speaker quite a bit during the presentation. Really need to emphasize what you are going to be discovering new. And from what I heard from the presentation is you're hoping to find new indigenous fungi from these sites. And that, if you were to actually go and do this work, it would be something new. And that's so I think we need to discuss that before. So, but, yeah.
All right, everybody, who's ready to be grossly underwhelmed with information?
I know.
All right,
It's not a good mix.
I got a lot of smiles out of that. That's audience interaction. I think we get started. All right, variable acid mine drainage conditions and their impacts on acidified bacillus rocks and iron precipitation. So, what is acid mine drainage? That is when sulfide minerals that have been dug up through mining processes have been exposed to water and air. That creates sulfuric acid and aqueous iron 2+. Basically, it's just this really gross solution that kills a lot of organic materials, especially aquatic organisms, plants, and it can contaminate things like drinking water. It can affect the plant life that is terrestrial around certain runoffs. It's a problem for rivers. It's a really, really nasty stuff, and it's attributed to a lot of different types of mining, but it's almost always from sulfide mineral related minings, commonly things like pyrite. It's not really used industrially. It's kind of a waste to get the iron out of pyrite because then you have to find a way to deal with all of the sulfur. So what they do is they take everything else out of the ores that they mine and they kind of just leave the pyrite alone when they're smelting it. And they put it in tailings piles, and we're going to get into that a little bit too. But they're really, really toxic. They're heavily acidic. They can range, well, so The average range that I've found is between 2 and 6, 2 and 5-ish, but they can be much lower. Some acid mine drainage sites can be sub 1 in terms of their pH levels. Dr. Ronkowitz, who no longer works at the university, actually found an acid mine drainage site that was like only 5 or something like that. It was really, really acidic. It also mobilizes other metals that isn't just iron. It can mobilize copper and zinc. It can also dissolve and mobilize heavy metals as well, which just further adds to the toxicity. Some concerns with mercury and lead, and actually we're going to get into the lead here in a minute. So acid mine drainage damages, well, like I said before, it kills aquatic life, it can contaminate your drinking water, mobilizes those heavy metals, so then those can cause further health issues for the surrounding terrestrial biota and then also humans if they come into contact and exposure with it. From the United States Forest Service, they predict that between 8,000 and 16,000 kilometers of streams have been affected by acid mine drainage in the United States alone. That's a lot of damaged area. And then also they have heavy costs to remediate. We're talking millions and millions of dollars to remediate small sites. Some sites that were in mining, I saw range between 120 to 150 million dollars to remediate a mine site. If you get it out in the environment, it's going to cost more because you got to go trace it down. You got to go chase it down and find it, which that's never good. And then also the other thing is it costs so much, these companies go belly up as a result. So then the taxpayers have to pay for it with Superfund sites and the national priority list, which, let's be honest, we don't like paying the government. I don't like paying the government. I'd rather not pay them for stuff like this if we can avoid it. So part of that is trying to find cheaper solutions. And so then I'm going to get into a specific area that we're looking at, Missouri lead mining. I know Jeffrey touched a little bit on this, but in Missouri we have really distinct metal compositions. So we're one of the only places in the world where we have a primary lead ore deposit with secondary zinc and tertiary copper. Normally lead and zinc are switched and is a primary zinc, secondary lead. So that is one of the reasons why Missouri has been like one of the lead capital of the world for 300 years or so. And so what ends up happening is they process all of this and then they put all of their materials that they don't use or after they've ground it and smelted it into tailings piles. So those are heavy in iron and sulfur chemicals because they don't need the sulfur and they don't need the iron at all. And so these leftovers get put in these tailing piles, which is then a prime location for acid mine drainage to start because they're exposed to the air, they're exposed to the elements, they have heavy iron sulfide presence. And so then one of the ways that these combat this is they create ponds to help contain the runoff in these basins that are controlled in the sense that they've tried to make them impermeable underneath to try and contain the runoff. But let's say we have a crazy storm or a flood, that's going to wash everything away and it's going to contaminate the surrounding area. So then, and this has been happening for hundreds of years. Missouri lead mining started in about 1700 when the French were here. So we're going to get to the organism now, acidified bacillus peroxidans. I'm just going to call it ATB. So it's an extremophilic acidophile. It's got an optimal growing pH between 1 and 2. 2 tends to be the most commonly used pH in terms of like its growth. It's present at a lot of AMD sites already, and it's used to remove the iron in its aqueous form from the solution. It uses oxygen with a terminal electron acceptor. It takes that electron from the iron and it gives it to the oxygens. And as a result, then it turns it into iron 3+, which is insoluble, and so then it precipitates out of the solution. And that's actually really important because lime treatment is one of the biggest ways you can treat the acidity problem in acid mine drainage. But with iron in the solution, lime treatment is less effective, and it takes more lime, and it doesn't do as good a job. And so then another couple interesting other things is that it fixes carbon and nitrogen from the atmosphere, which is pretty neat. So it's pretty self-sustaining as well. You don't have to add a ton of materials in terms of like organic carbon to your site to encourage the growth. And it's got an optimal temperature of about 30 degrees Celsius in terms of its growth, which is pretty doable for a lot of environments and aspects. So the experiment, we're going to use ATB to precipitate iron out of an acid mine drainage solution in conjunction with two variables. We're going to vary the lead concentration, and that concentration is not the total concentration of the solution, it is the percentage of the metals in the solution, so it's a relative percentage compared to how much metal is there. And then we're also going to vary the pH. So the hypotheses that I'm working on here, the pH of 2 with a lead of 0 is going to be the most effective at iron precipitation, but lower pHs will have a bigger impact when there is higher lead. So if you have lower pHs, it's going to encourage these organisms to precipitate the iron out even with higher lead percentages. I'm estimating 1.5 or 2 is probably going to be the most successful range for that, because that is a kind of optimal pH range. So bringing in culturing, collection. We're going to locate an acid mine drainage site not contaminated with lead. Reason being, I want to prevent prior lead exposure. I don't want to grow organisms that are already resistant to lead. That kind of defeats the purpose of what we're doing here. And so by doing that, we're going to have a clean sample. So then they're not going to have prior resistances to lead. So then our experiment is going to run with what these organisms can do with the introduction of lead as opposed to with the prior exposure of lead. Then we're going to use ethanol sterilized shovels and buckets. We're going to take samples from the bank surrounding the acid mine drainage, like pond or area in which it's been affected, and we're going to take several samples. It depends on kind of the site. If you're looking at kind of a tailings pond, you would take a couple samples from the rim of the tailings pond that's really close to the edge of the water, and therefore that's going to be the area where you're going to find the most amount of activity from the ATVs. So now we're going to go into identification. So we're going to take those soil samples and we're going to use sonic separation and a sieve to sort out all of the sediments to try and get the microbes. Then we're going to introduce the microbes to a solution. It's going to have a pH of 1 because not a lot of stuff can survive at pH 1, but ATV can. Then it's going to have a high iron 2 aqueous solution. That way it can kind of like start growing them a little better. And then we're going to use an agar streak to isolate the communities. When we isolate the communities, we're going to run 16S genomic sequencing on the communities. So we can actually isolate the ATV as compared to other acidophiles that might be present. Then we're going to get into enriching. So when we enrich it, we're going to use a solution of a pH of 2 that is a sulfuric acid base, because they're really attuned to high sulfuric acid content solutions. The temperature is going to be 30 C. That's going to be very high in iron 2 slugs in the aqueous form. Then we're going to add glass microbeads. ATV works really well when it's immobilized on some sort of solid media. And as a result, then to count them, we're going to use a scanning electron microscope, and we are going to count what is on the surface per unit area. And then we're going to extrapolate that out to the rest of the beads that we have added to the solution. And then with that, we're going to try and shoot for a magnitude of 10 to the 6th microbes present as our goal for saying that this enrichment culture has succeeded and can proceed to the experiment. And we're going to need 50 cultures. I have 5 pH levels, I have 5 lead percentages, and we're running duplicates for accuracy's sake. So the pH levels are going to vary. It's going to be 1 to 3 in increments of 0.5. The lead concentration is going to vary from 0 to 4. It's going to go to half, and it's going to double. So 0, half, 1, 2, 4. We're going to use a sulfuric acid base again to imitate acid mine drainage. It's going to have 2% copper and 2% zinc in the solution. That is to imitate the Missouri lead tailings and the area, because those are minerals that are found in our mining areas. And then the iron 2 plus aqueous, in terms of its relative percentage of the metals, is going to vary as well, depending on how much lead there is. Since you can only have 100%, it's going to vary between 96 and 92%, just to account for the differences in lead. So now we're going to talk about the collection from the experiment. So you're going to collect aliquots from the sample at 5-day intervals, starting at 0, going to 45. And so what's going to end up happening then is you're going to sieve out those samples from the solids, because you're going to have precipitated iron 3+, and you're also going to have glass microbeads in this solution. So you're going to just want to remove the solids and return those to the vessel so they can continue contributing to the experiment. And then you're going to take that aliquot that you've now sieved out, and you're going to use atomic absorption spectrometry. We haven't really talked about this in our class. There's a picture of how it works. Basically, you take your solution and it burns it, and then it tells you what's there in terms of like parts per million, parts per billion. And so why that's important is a lot of other methods aren't capable of telling you good percentages or quantities of aqueous metals in solution. That's really what we're looking for. And so we're going to analyze the aqueous metals that are present in this solution. We're not really expecting a change in the copper and in the zinc or the lead, but we are expecting changes in the iron in terms of its parts per million and parts per billion. And so then we can use that then to mathematically determine the relative percentage of the iron to in terms of the other metals, and hopefully it can precipitate enough out where that number starts lowering rather drastically. So the expected results, the lower pHs allow for more efficient iron precipitation at higher lead levels, expecting the optimal pH to get 2, but with 1.5 and 1 to be more efficient at this than, say, a pH of 3. And then lead will slow down the microbial precipitation, and I'm thinking that it's going to have an increase in the fact that higher percentages of lead does affect living organisms, and it can slow down microbes in their endeavors. Then the applications for this, if we can remove the aqueous iron 2 plus from solution, the lime is going to work better. And then also, through a lot of recovery methods, iron can get in the way of that. And if you want to, say, take this solution and for economic purpose and separate out the copper and the zinc from it, removing the iron will allow that to be more efficient and to work better. And it will allow the recovery of those other metals from the solution. Then you can also apply this to lead mining sites. A lot of these are applied to things like iron mining sites and other mines where iron is heavy. You can apply these to tailings or runoff areas. And then also it's going to assist in cost reduction for remediation as well, because this is a much cheaper method than dumping thousands of square kilometers of lime in an area and having it not work as efficiently. Does anybody have any questions? I can probably answer some of them.
Josh. Okay, I've got a couple of questions.
Okay.
Of course. So, well, one, with the acidophilobacillus, actually, I don't know. And I am curious. So, has anybody actually done some studies to determine how tolerant these organisms are to things like lead?
Not that I've seen, but I've been looking, trying to find things related. I haven't seen much, but it's possible. So I'm varying the lead and the pH at the same time to try and create a unique study as well. By varying the two, I suppose that I can create something that other people haven't to try and, it's well known that microbes are affected by lead, and it can slow them down quite a bit. And so by varying pHs within its acceptable range, perhaps we can find a pH that is optimal, so that at your site, let's say we have a pH of 3, it might actually behoove you to lower your pH to 2 to enable their function better if there is the presence of lead. So by doing this, I can apply a different knowledge to that.
Well, and then also with the metabolism of this organism, it's naturally going to lower the pH anyway. So, which raises another question. So with your test, or your assay itself, you probably would have to think about buffering to make sure that you maintain your set pHs throughout your experiment, because these organisms will alter their environment. Let's see. Another question I have is, well, you go and you get these environmental samples. What happens if you don't get these organisms isolated from those samples?
Go back out and try again. I have an infinite budget and infinite time.
Okay,
Go to a
yes,
different
I
site
did say
and try
that.
again.
Okay. Okay, so, and I guess my final question is, since you sort of want organisms that are going to have some lead tolerance, why don't you start off with the actual environmental samples that have been exposed to lead? And
I would like to see what happens when they haven't been exposed to lead, perhaps as it could pertain to a new introduced sample of lead in the area, see if they're still functional. I was more because if they have lead resistance, they're going to work better. I want to see what happens when they don't work as well as they should without lead resistance. I felt like that would just kind of add a little bit more to the knowledge of the organism.
okay, yeah, I don't actually think you need that so much. because, one, through doing this particular set of experiments, one, you are going to determine the tolerance of bacteria to lead. And it would be actually, and the thing is that if you're treating areas, especially specific to these Missouri sites, you probably want to use things that are more indigenous to the site, because I think that to get away from the possible lead contamination, yeah, you'd have to go much further away. Missouri. So I guess what I'm saying is, I don't think you necessarily have to try and isolate the organisms from non-lead contaminated sites. I think you can actually be successful with pulling organisms from around the sites that you want to treat.
Would it be possible to take both and then just incorporate both of them and run it two at a time?
Yeah.
I can have 100 cultures because I have all the money.
You have all the money. You can hire all the technicians.
Okay, cool. What's that?
Hire a bunch of undergrad minions.
That's right.
I've got infinite budget, infinite time. This is going to be great.
Okay. Any other questions for Josh?
Would any of your applications be able to be used on zombies?
You see, probably not.
Okay,
Unless
okay.
you wanted to take a less acidic solution, create a more acidic solution to then burn these zombies with acid.
Acid will burn them, but not the flamethrower. I'm just throwing it out there that that's a thing.
It's not going to eat through the road and it's going to burn down a house.
That's fair.
Okay, I don't think we have to worry about doing bioremediation of zombies.
When it happens, don't come knocking on my door. I'm just
Acids
saying.
work kind of slow though, so I don't know if it'd be efficient at
Don't
killing
say.
zombies.
Okay.
Like you don't have time to like get rid of the acid.
It's a zombie, isn't it?
Valid.
That
That's
can
valid.
go a while. Did I succeed in the goal that I've set out to achieve?
You gave a pretty good presentation. I don't know what you're like.
Okay, let's thank Brenna, Ben, and Zara. Even with the time crunch, we're getting out of here a little bit early. So Friday, Peter and Abdul. So looking forward to your presentations. So see you guys on